Hi, everyone. I'm Mason Zhang, a rising junior at Brown University studying computer science and a summer trainee in the Mayan lab at the Icahn School of Medicine at Mount Sinai. Today, I'll be presenting on creating an age prediction model based on RNA-seq profiling of peripheral blood mononuclear cells, otherwise known as PBMCs. PBMCs are white blood cells located in peripheral blood. In two previous studies done by the Mayan lab, Clark et al. Developed a machine learning classifier trained on data from PBMCs profiled with RNA-seq to diagnose Lyme disease. In a more recent study that also involved members of the Mayan lab, data from PBMCs treated with plasma from patients with atherosclerosis, also profiled with RNA-seq, was used to identify anti-inflammatory drugs. My project also uses PBMCs, but for a different task, age prediction. Aging clocks are machine learning models that predict biological age. Horvath was the first to implement this approach using omics datasets. He used DNA metabolomics in particular to predict the biological age of patients with great accuracy. In a related study led by Zichin Wang at the Mayan lab, an aging clock was developed by processing data from over 1 million electronic medical records. Vital signs and lab tests were used as features to train a deep learning model that could be used to predict biological age. The figure on the right is from that paper. It shows that the model had a mean error of 7.6 years. Another important ingredient of this project is ArchS4. ArchS4 is a Mayan lab resource that was mainly developed by Dr. Alex Lachman, a research assistant professor in the lab. ArchS4 provides all the RNA-seq data from the gene expression omnibus uniformly aligned. From ArchS4, I received from Alex a collection of around 5,000 RNA-seq samples collected from PBMCs. An important aspect about these samples is that each sample had the age of the patient. Now I was ready to construct my own aging clock. Here is a brief overview of the workflow. The model takes in RNA-seq data and clinical metadata extracted from ArchS4. Then outliers are filtered out through dimensionality reduction and clustering. Different models were tested for model selection, and we ultimately decided to settle on using LightGBM based on its performance. Batch correction and different normalization methods were also tested, but ultimately none were used. The RNA-seq data was then inputted into the model for training and testing through cross-validation. At the same time, after training, the most predictive genes were queried from common biological functions of Enricher, and the model was tested on holdout series. Next, I'll go in more detail to explain the methods and discuss the results. First, for removing outliers, I used principal component analysis to visualize all samples in 2 dimensions. This analysis enabled the identification and removal of outlier samples and studies. The original RNA-seq data contained over 5,000 samples from 98 studies, but after removing samples from ages younger than 20, samples that isolated and sorted specific cells before profiling, samples from single-cell RNA-seq, and samples collected from other cell types, 1,400 samples from 35 studies remained for training and testing the model. Now, originally, there were a total of 62,000 genes in each RNA-seq sample. To keep the model simpler and more interpretable, we decided to only keep protein coding genes. This reduced the number of genes to around 19,000. Next, we wanted to check how many genes we actually need to make accurate predictions. As the plot shows, the error decreases dramatically when the model is trained with the top 10 to top 250 genes, but then the error decreases slower until it plateaus when more genes are included. Although all the protein coding genes were eventually used in the model, only around 2,000 of them had feature importances greater than 0. To train the model, the LightGBM regressor was trained using 90% of the data, and the rest of the samples were used for testing. The trained model had a mean absolute error of 6.8 years and a mean absolute percentage error of 15% of the testing set. From the line of best fit, one can observe that the model had some variation, but overall had a strong positive trend. Some of the error may also be due to real variance, where a patient's chronological age may not be reflective of their biological age. To further assess the model was trained on all series, but one was held out for testing. This procedure was repeated for all 35 studies. Overall, the mean Pearson correlation was 0.317, and the maximum was 0.878. This suggests that the model does not overfit, but is also not fully generalizable. In the histogram on the bottom left, most of the holdout series are concentrated on the right positive side. The negative correlations were mostly due to either sample size being too small, for example, one study only having 3 samples, or age range being limited to 1 or few years. Also, it is important to note that the model performed worse on older ages, and this is likely due to having less data from those age ranges, as shown by the plots on the right. Next, I will discuss the contribution of genes to model prediction. The model's ability to identify age-related genes was assessed by comparing the most predictive genes to gene sets known to be associated with aging. For instance, 160 out of the top 1,000 predictive genes were identified to be previously age-related by the Human Aging Genomics Resources database. XenoRanger, GenAge, and LongevityMap also provided gene sets that we could use to compare. Based on the Fisher exact test, the overlap between the model's top 1,000 genes and the genes in LongevityMap and XenoRanger are significant, but the overlap with GenAge was not. Although the overlap seems to be small, the model significantly picked age-related genes as the top features. The additional top feature genes might be predictions of genes not yet to be discovered to be related to aging. Moreover, I used enrichment analysis to examine the biological processes associated with the most predictive genes. First, these genes were divided into those that either positively or negatively correlate with age. It appears that cellular response to UVB is enriched for genes positively correlated with age. UVB is associated with apoptosis and premature senescence in skin cells, which might point to similar effects in blood cells. Meanwhile, positive regulation of T helper 17 cell differentiation is enriched for genes that are negatively correlated with age. This is pointing to the known increase in inflammation with aging. Another goal was to identify novel genes. Only 2 genes were used by all the holdout models in more than 15% of its decision trees. These genes are NRCam and Fidgetin. These 2 genes have not been previously identified as age-related. NRCam was the most important feature in the model. Knockout mice of NRCam had phenotypes such as decreased grip strength, impaired coordination, and other motor neuronal-related phenotypes observed in the elderly. Fidgetin is also known to be associated with mitochondrial dysfunction and DNA damage, possibly explaining its importance in the model. Overall, I was able to produce a new aging clock using publicly available RNA-seq data collected from PBMCs. Through benchmarking with known aging gene sets, the model can identify age-related genes. To improve the model's accuracy, we could potentially use additional RNA-seq data from other sources, for example, GTEx. I would first like to thank Dr. Mayan and Ms. Jenkins for their support. I also want to express my gratitude to my collaborators on this project, Alex, Nash, and Daniel. Finally, I would like to thank all members of the Mayan Lab and my fellow summer trainees. Thank you for listening to my presentation. Please put any questions in the Q&A feature. The next speaker will be Ria Desai, who will be presenting on her project analyzing trends in bioinformatics tool publication.